Welcome to another video. In the previous video, we were discuss about the tissue culture, what is the tissue culture, and growth medium. In this lecture, we will discuss about the growth regulators. So, growth regulators means to improve or to growth, to produce the growth in the plant, the chemical which are responsible for production of growth. So, auxin produces growth in the root and cytokinin produces growth inside the shoot, means auxin increase the root and cytokinin responsible for increasing shoot. So gibberellin also cell enlargement, and abscisic acid, which is the plant trace hormones, also we are studying in the growth regulators because they are responsible for increasing the fruit length. And so ethylene is very bad. So auxin, how many auxins? The IAA, IBA, NAA means indole acetic acid, indole butyric acid, naphthalic acetic acid, and 2,4-D. These are well known weedicides, all are known about that. 2,4,5-T and picloram. So auxin is the callus formation, rooting of cuttings, and the induction of adventitious embryo. So callus formation, callus is the undifferentiated mass of cells which are not differentiated into root or shoot. This is also a very laboratory condition you can observe in the vessels or bottles. The rooting of cuttings means you are sowing the roots by cutting. Cutting method is the vegetatively propagated methods. So auxins are used inside the rooting of cutting and induction of advantageous embryo. So cytokinin, the cytokinin which is the enhanced advantageous shoot formation. Example is the BA, means benzyl aminopurine, then 2 IP, 2 inorganic phosphates, then kinetin and zeatin. This is the example of the cytokinins. And gibberellin, not generally used in the tissue culture, tends to suppress root formation and advantageous embryo formation. So gibberellin isolated from the one fungi. The name of this fungi is the Gibberellia fusocoria, first occur inside the Japan, also called as the foolish disease. And the abscisic acid is the primarily a growth inhibitor but enables more normal development of embryos, both zygotic and advantageous. So ethylene, the question is not how much to add, but how to get rid of its in vitro. In vitro means in the laboratory condition. So the natural substances produced by the tissue culture at fairly high level, especially when cells are under stressed. Then enhance senescence suppression. Enhance of the senescence. Senescence is the leaf fall down, and suppress embryogenesis and development in general. Embryogenesis means genesis or embryo formation in the tissue culture condition. Then hormones combination. In the tissue culture, you want to develop the callus, so you must combine the hormones. The advantageous embryogenesis, then next is the rooting of shooting cuttings, then advantageous shoot and root formation with the combination of hormones. Okay, so you can see here in this table a selected list of the plant growth regulators used in the culture media. This is the auxins and this is the cytokinins. The IAA, IBA, NAA, 2,4-D, etc. All this is the auxins example of the growth hormones, and cytokinin BAP, benzyl aminopurine, benzylamines, then DUP, kinetin, zeatin, then thiodiazorone. This is the different types of the growth hormones or growth regulators. The chemical names: indole 3-butyric acetic acid, indole 3-butyric acid. Then 1 naphthalenic acid, 2,4 dichlorophenoxy acetic acid, 2,4,5 trichlorophenoxy acetic acid. So 4 chlorophenoxy acetic acid, then nephaloxy acetic acid, then methyl 4 chlorophenoxy acetic acid, and 2 methoxy 3 6 dichlorobenzoic acid, and picoloram is the 4 amino 2,4,6 trichloropiconolic acid. Then BAP 6 benzyl aminopurine, benzyl adenine, then N6 2 isopentyl adenine, then DUP means diphenylurea, then kinetic 6 furfuryl aminopurine, G13 means 4 hydroxy 3 methyl trans 2 butyl aminopurine, and thiazurone is the 1 phenyl or 1,2,3 thiazole phyl urea. This is the name of the growth hormones and classification: auxin and cytokinase. So the between combination of the growth hormones we develop in the in vitro condition the callus. You can see here the photograph of the callus. Callus means undifferentiated mass of cell. So it is an unspecialized, unorganized, growing and dividing mass of cell. So it produces when explant are cultured on the approximate salt or solid medium with both an auxin and cytokinin in an correct condition. 2,4-D are commonly used. 2,4-D is the well-known weedicide, and this also enhance the cell division. So that's why in the callus culture first 2,4-D is the commonly used and giving good result about the callus, and you can observe here. So during callus formation there is some degree of de-differentiation both in morphology and metabolism, resulting in the loss the ability of the photosynthesis. You can see here the color of callus. The callus, which is brown in color, not in green in color, means de-differentiation, means it is a loss of ability to photosynthesis, means photosynthetic pigment, which is the green pigment, totally absent here. So a callus is the blob of tissue. You can see here the naturally occurring callus in the nature. You can observe here the photograph shows the naturally occurring, the mostly undifferentiated cells. The cells is naturally developed on the plant as a result of the wound. When we are wounding to the plants, they produce the new cells or new tissue. This is the callus. So this callus can be left to develop or can be further divided. Okay, so callus culture may be compact or friable. So you can see here the friable, and you can see here the callus is the compact. So compact cells shows densely aggregated cells, means compactly arranged cells occur inside the compact cells. Then friable callus shows loosely associated cells, and the callus becomes soft and break apart easily, means this is very breakable or easily breakable. When you touch to the callus is break. Then habituations, it is reduce the requirement of auxin or cytokinin by the culture during long term culture. Then properties of the callus. So it often comes from shoot in early culturing. It can be maintained indefinitely, then no photosynthesis, and grows in dark condition. It can be used to isolate single cell with stem like properties. This is the totipotency. In the previous lecture we are observing the what is the mean by totipotency, means potentiality of the cell to grow new plant. So 3 stages of the callus. First, the introduction, cell is the explant, de-differentiated and begins to divide. Then second is the proliferative stage, rapid cell division means fast cell division and produce blob. then a morphogenesis stage, and differentiation and formation of organized structure, specially process that leads to plant regeneration from somatic cells. Somatic cells, which are the capacity to regrow. So you can see here in the medium or tissue culture medium or laboratory condition, the single cell clones. The plant body is made up of wide range of the innumerable cells that type are successfully integrated both in terms of the structure and function. So this is the single cell culture. So this is the cell tissue. You can take from the tissue the single cell culture on the culture medium. So single cell culture is the method of growing isolate single cell aseptically means in the laboratory condition, laminar airflow, all these things are used on a nutrient medium under controlled condition. Okay, so this is the single cell culture. In principle of single cell culture, why we are using the single cell? Because the basic principle of single cell culture is the isolation of large number of intact living cells and culture them on the suitable nutrient medium for their requisite growth and development. So requisite growth and development and suitable nutrient medium used a single cell. So principle of single cell culture, the another single cell can be isolated from a variety of tissue, means different tissue are present on the plant or organs or green plant as well as from callus tissue and cell suspension, the single cell from the intact plant tissue such as the leaf, stem and root are isolated either mechanically or enzymatically. So single cell isolated from the plant in the genetic recombination when you are putting the DNA in some cells. So in that condition the single cell is the most important because these cells contain the foreign DNA which are our experiment. So these cells isolated from the mechanically or enzymatically. Mechanically means in the under the observation of microscope with the help of some needles and forceps, or enzymatically means giving the treatment to the enzymes to the tissue and isolated from tissue a single cell. So this is the methods. So method of single cell culture, the first, the 5 important methods are widely employed for culturing single cells. So paper wrapped nurse technique, then petri dish plating techniques, then micro chamber techniques, then nurse callus techniques. This is the micro droplet techniques. These all are the different type of techniques used for the isolation of single cells. So importance of single cell, the importance of the fundamental and mutation studies. Okay, it has wide industries applications, means in the some seed industries and many industries isolated single cell for giving the mutation treatment to the seeds or plants. The single cell culture could be used successfully to obtain single cell clone. So plant could be regenerated from the callus tissue, means importance of the single cells. The single cell inoculate inside the media, this single cell divides and produce a callus, and this callus produce a purely those you want to mutation or cell what is the genetically engineered. The single cell culture are very important in relationship to crop improvement programs, means if you want to insert some cells inside the new crop you can use the single cell. So the importance of single cell, the single cell culture in the large scale could become a valuable techniques for industrial production of such important natural compound. This is the chemical purpose. Then polyploidy can easily be achieved by single cell cultures. The next is the organogenesis. Organogenesis means production of organs in vitro or in tissue culture. Different type of organs of the plant such as the leaf, root, stem, flower, all these organs in produce. The organogenesis is the process of differentiation by which plant organ with roots, shoot, bud, flower, stem, etc. are formed, while advantageous refers to the development of organ or embryo from unusual point of origin of an organized explant where a performed meristematic is lacking. So this process is much more common than somatic embryogenesis. Somatic embryogenesis we will discuss in next slides, and has for more potential for mass clonal propagation of the plant. This is the organogenesis: shoot production and root production, then rooting and shooting. The finally whole plant will be develop. The stages of organogenesis: this is the first 2 3 4 5 and 6. This is the shoot tip in the medium. You can see shoot tip, the first shoot callus, then third is the shoot of stem. You can see here all the shoot, the green shoots are occurs in the inside the medium. Then roots, the roots are occur inside the medium. Then hardening is the process where the laboratory condition will be finished, and the next stage, the small plantlets are sowing inside some small bags or in the shade house. This is the, and the final is the whole plant growth. This is the stages of development of organogenesis. All organs are developed in these stages. So what is meant by somatic embryogenesis? And somatic embryogenesis is the process by which somatic cells of the cell tissue, including haploid cells, develop into differentiated embryos and to regenerate plants. Then Shetworth et al., the name of the scientist, 1958, first induced embryo through suspension culture in carrot. So Reinert, 1959, produced embryo from callus in the carrot through suspension culture. So what is the importance of somatic embryogenesis? The first is the higher propagation rate, then suitable in suspension culture, then artificial seed production, the labor savings. This all is the importance of the somatic embryogenesis. So you can see here the stages of somatic embryogenesis. The first, single cell to from heart shaped embryogenesis. You can see cell division. This is the single cell division produce the callus, then again single cells, and finally heart stage or heart shaped stage, and tripod stage, and finally plant. So this is the stages of development of the embryogenesis. Then types of somatic embryogenesis. So there are two type of somatic embryogenesis: direct and indirect. In this direct, the first, the embryo initiate directly from explant. In the absence of callus formation, embryo are formed due to pedicels, means pre-embryo determined cells. And the indirect somatic embryogenesis means callus from explant take place, from which embryo are developed, and embryo are formed due to induced embryonic determined cells. So this is the two method to development of the somatic cells or somatic embryo. So embryo culture. So isolation of growth of an immature or mature embryo in vitro with the goal of obtaining a viable plants. And the first attempt to grow the embryo of angiosperms was made by Henning 1904 from two cruciferous cochlearia and Rapnus. This is the types of embryo cultures: mature embryo culture and immature embryo culture. The mature embryo culture, it is the culture of mature embryo, so derived from the ripe seeds. This type of culture is done when embryo do not survive in vivo. In vivo means in natural condition, or become a dormant for long periods of the time. So this culture is done to eliminate the inhibition of seed generation. And immature embryo culture, in that situation, or in this case of the immature, it is the culture of immature embryo to rescue the embryo of wide crosses, means you can cross the two plants. So in that situation, so immature embryo used, because cross will not giving to the pure variety, so you can use immature embryo. This type of culture is mainly used to avoid embryo abortions with the purpose of the production of viable plants. So media requirements. So for mature embryo, basal salt medium with a carbon energy source. For immature embryo, different vitamin, amino acid and growth regulators are required. Then culture mediums, the mineral salts such as the potassium, calcium, nitrogen, most important, the carbohydrate and most pressure. So 2% sucrose works well for mature embryo and 8 to 12% of the immature embryo. Then transfer to progressively lower level as embryo growth. So culture medium factors, the amino acids reduces. N means nitrogen is often helpful. Up to 10 amino acid can be added to replace N salt or nitrogen salt, including glutamine, alanine, arginine, aspartic acid, etc. Require filter steriling a portion of the medium. Then natural plant extracts such as the coconut milk, liquid endosperm of the coconut, then enhance growth attributed to underfined hormonal factors and or organic compounds. The other extract of dates, bananas, milk, tomato juice you can use it in the culture medium. The plant growth regulators, the globular embryo require low concentration of auxin and cytokinin for growth of the root and shoot. The hurt stage and later moderate level of the auxin and low level of the cytokinin. So gibberellic acid and abscisic acid regulate precocious germination.